Ushma Neal here with the next in the JCI series, Conversations with Giants in Medicine. I have the distinct pleasure of speaking today with my old friend Napoleon Ferrara of the University of California, San Diego. Napoleon is best known for isolating and cloning VEGF, the vascular endothelial growth factor. He then built the humanized monoclonal antibody that led to the blockbuster drugs Avastin for treating cancer and Lucentis for treating wet age-related macular degeneration. His research has always been at the cutting edge of angiogenesis, and I look forward to hearing a little bit more about his unique path through science today. Thank you for joining me today, Napo.
It's a great pleasure.
Do you think you could start at the very beginning? Where did you grow up? What were your parents like? What were you like as a kid?
Yeah, well, I grew up in a city called Catania, which is in Sicily, in southern Italy. Actually, it was a very nice city with a beautiful geographic setting. We're next, to Mount Etna, which is the highest volcano in Europe, and then the beautiful Mediterranean Sea. So the city is a pretty place. And my parents were, my father was a judge, and my mother was actually, she has a teaching credential, even though she really never taught, and I was fascinated, by law. My father took me to court, a number of times, and so did his brother. My uncle was a criminal lawyer. Even though I thought it was very interesting, it was never enough really to think, oh, I want to be a lawyer or a judge. The person, probably the great influence on me was my maternal grandfather. Was actually a science teacher. I still remember this house full of books, probably thousands of books. I was so fascinated by those. Sometimes I believe I stole some of these books, even though then I returned all of them, And I think that was really what began me an interest in science.
Was it natural science, chemistry, or medicine? What was the early inspiration that led you to medical school?
It was mostly natural science, actually. It was a teacher, it was a high school teacher of natural sciences. He did an enormous collection of books. He was a bibliophile. His library would be at home, maybe with 5,000 books. So that was his real passion, which a little bit, transmitted on me as well.
So after reading all of these natural science books, what then led you to choose to go to medical school?
Well, because at that time, at least in Italy, medical school seems to be probably the most, logical path, even if you want to do research. At that time, remember, there was not even well-defined, PhD program, in Italy. I think they started a PhD program at the same time or about the same time that, I finished, medical school. So it was a pretty logical path, to go to take medical school, and then you decide what you do next. And this is pretty much what happened to me. For a number of years, I was not totally sure what I really want to do, but and medical school, I guess, was a great choice because it provided a biological background, which I think has been extremely helpful even in guiding my research to ask some questions which I thought to be medical and biologically relevant.
So we'll come back to your research for a second, but you ended up choosing to do your training in reproductive endocrinology. Why?
Because at the time it was it was a sort of a cutting edge, science. It was a very fascinating work. In this, I think, it was very much inspired, influenced by the person I consider my first, mentor was Dr. Scapagnini, Umberto Scapagnini. He was a young, professor of pharmacology. He was a full professor at 34 or at 35, and very charismatic personality. He spent, several years in the States. Actually, at the UCSF, he worked with William Ganong, was one of the giants in endocrinology, neuroendocrinology. And he brought, this, I would say, University of Catania was a very good, medical school, but mostly clinical oriented. There was not a lot of research going on, at least at that time. And Scapagnini brought, this excitement for research. I still remember vividly his lecture explaining, the neurotransmitter, the hypothalamic releasing and inhibitory factor, the pituitary portal system. I thought it was so intriguing, so fascinating that this is what I really wanted, to do. And that what really led me to the path of research.
So you did do some research with Scapagnini during...
A little bit. As a medical student, I did a little bit of research.
Okay. But then you set sail after your clinical training for UCSF to Richard Weiner's lab.
Yes, Richard Weiner's lab was essentially, it was a continuity, with what I was doing with Umberto Scapagnini. Richard Weiner was working in neuroendocrinology. Actually, he was a good friend of Scapagnini. He's the one who certainly helped me to get this opportunity with this fellowship at the UCSF. And I found the work in an incredibly fascinating topic, which was the pituitary gland. I think at the beginning what we were looking for something completely different. But I kind of stumble in some cells, I try to culture some bovine pituitary. I came across, these cells, which called follicular stellate cells. Actually, it took a while to identify the cells. There were some epithelial cells which form some domes, which is reflect epithelial transport, which was a very surprising because nobody associated pituitary cells with iron and water transport. Has been mostly associated with the kidney epithelial cells. So at the time it was a very pretty interesting discovery. I spent countless hours, weekends, try to decipher, try to interpret what these cells were doing. And in the course of the studies, I actually had this idea, could have been a crazy idea, to test the condition media of the cells, on endothelial cells. There was actually some early hypotheses which suggested follicular cells, among maybe a dozen of other hypotheses, play a role in the organization of the very complex pituitary vasculature, because they very frequently associated with blood vessels. At the time, to my delight, to my surprise, there was a very strong mitogenic effect on endothelial cells. So that's what really started the VEGF story, which actually occupied me for the last, I would say, since maybe that must have been 1984. So we're talking about over 30 years.
Yeah, right. Is it true that you had to go to slaughterhouses to get cow pituitary glands?
I did many times. Even Dr. Weiners and I came to the slaughterhouse, at least to show me how to collect, pituitaries. There was nothing wrong in doing that. I thought it was you have the full circle of doing research. You start with collecting your tissue, your specimen, and then you process them and you culture them.
Now, when you had this aha moment of putting the conditioned media on endothelial cells, well, let me ask the question a different way. Was there an aha moment when you saw that the endothelial cells started dividing with the conditioned media?
Well, I think there was many aha moments, because this was very preliminary. It was exciting, but even people around me were telling me does not necessarily mean a lot. Could be one of the many known factors. You should not make a big deal out of that, etc. And I understood that at that time. Also follow up this observation could have been extremely difficult. Those are the days of the pre-genomic days, when to find a factor. There was an incredibly laborious process, to isolate a protein or purify it, to homogenate. That could take a decade. And so many people discouraged me from pursuing all this story. My guess, I still persisted.
Well, we're all happy that you did, but
Yeah.
At that time, did you ever consider a clinical career? Did you ever consider going back to Italy? What kept you on this track?
I think I did. This is a great question, because at that time I was really, I was thinking that combining research and clinical work would have been really probably the logical path. And actually I interrupted my research career. Actually, I started residency at Oregon Health Science University, actually in OBGYN, actually, and thinking that continuing doing a full residence and then the fellowship was the thing to do. There is lots of people around me were telling me that. And so I began my residency. That was 1985. And I must say that while I appreciate the clinical background, I missed the lab a lot. I was very unhappy doing this period. And then I decided to go back to UCSF to do actually a second fellowship, research fellowship. And I think it was actually the very important experience for me, because it really guided me, made me understand what I want to do and what I didn't want to do.
Why not return to Italy then? Why choose the place where you rested for about 25 years, which was Genentech, which was in the Bay Area?
Well, Italy is a wonderful country, but I mean, doing some work is a bit, complicated. Doing some work, there was a lots of difficulty even in an academic career. So while I consider that, but in reality, I thought, staying in the United States could be probably would have facilitated, this work. I know that there are some people who in Italy, one of the European countries, have done extremely well, but I felt that probably United States was a good place to continue my research. And so after my second fellowship was actually in the Cancer Research Institute. Actually, was a lab in which was focused on growth factors, so provide me a great experience, a great training in that. I was looking at a number of opportunities. I was very fortunate actually to have an opportunity to join Genentech. Maybe in those days was not exactly the thing to do because usually academia was the ideal place. I understood that this was the right place to continue this work.
What kept you there for 25 years?
Well, because the truth, it turned out to be a great place, because actually there was a fantastic technology, fantastic colleagues, and I thought I had the opportunity a number of times, but then I concluded that Genentech was the best place to be. I still, now I'm at UCSD. In the mid-90s, I had a great opportunity joining UCSF. I always thought, I have a great respect for academia. And so I had, this opportunity to join the UCSF. At the time, there was this newly formed, cancer center, and it was an extremely tempting offer. And I debate a lot. It was a very difficult choice. But then I concluded, probably staying at Genentech would be, at that time, in the mid-90s, would have been the best place to continue my research. I think it was a very good choice at that time, because it's totally possible that this work on angiogenesis, on VEGF, Avastin, Lucentis, maybe would never taken place.
All right, let's rewind a little bit. Your first project that you were assigned to at Genentech drew on your reproductive endocrinology and OBGYN training, as you were assigned to work on relaxin, which was thought to potentially ease the pain of childbirth. So how did that project go?
Oh, this was a great project, relaxin. It was a hormone which has been shown in mice to have a dramatic effect, plays a very important role in facilitating parturition, because it dilates the pubic symphysis. There was actually a dramatic effect in mice. And so many people speculated the same could be true in humans. But the truth of the biology was very, very different. Myself and many other people spent, years, trying to reproduce, try to make a relaxin work, in not only in other rodent species, but in primates. But, the data was extremely inconsistent, And in retrospect now we know that that was really the wrong biology, One should be very humble and try to understand that what happens in mice may not be true in humans. There is an enormous difference, So we spend a lot of time, trying to make relaxin works. But once again, we're unable to do that. They even did some clinical trial with the relaxin, in humans. It failed. It did almost nothing. And what is particularly intriguing, now that relaxin has been developed as a drug, for congestive heart failure, something completely different, which is really show how fascinating and unpredictable biology can be. It shows some promising results in that.
Wow. During that time, you started working on VEGF as a side project.
Yeah.
Genentech let you just have time to work on something you weren't otherwise assigned to?
This is one of really the things that I remember with the greatest fondness, those early days when I joined Genentech. Yes, my main project was the relaxin project, but I think I quickly understood that this project was really was very unlikely to work. My real passion was to continue the work that I began at UCSF with this mysterious protein, because at the time we didn't know what it was. Could have been anything. It could have been one of the previously known growth factors. So I worked extremely. I spend, really many hours, all my weekends, try to purify this protein, to isolate, this protein. I was very fortunate to find some incredibly talented, colleagues and collaborators. One I would remember was Bill Hensel, was really an expert in protein sequencing, in one of those people who pushed the technology to the extent that with the very extremely small amounts of protein you could obtain a sequence. And it was almost miraculous because in almost a little over 6 months after I joined Genentech, we've been able, to fully purify and have an N-terminal sequence of VEGF, from follicular stellate cells. That was probably the most exciting moment when actually we compare, the sequence with the database. At that time, we did not have any match in the database. At that time, it was a novel sequence. That was a really very, very exciting time indeed.
So it seems to have captivated you for the last 30 years.
Yes.
The sequence, the structure, the receptors, knocking things out in terms of the genetics, and then later on as a therapeutic
Yes.
target. So what is it about this one molecule that seems to have enraptured you for 30 years?
Well, I feel immensely fortunate that actually I came across, this molecule because it proved to be a very important molecule. It was very fruitful, spending, a few decades, studying this molecule. I've always have a kind of, an inclination to take some risky, project, side, direction, and away from VEGF. Each time that I did that, I realized that I was going to miss some very important discovery. So all of this kept me back, to work on VEGF, and I believe it's been a very, very fruitful, very fruitful research.
Do you think there's more to find out about VEGF and the way that it acts?
Well, I think VEGF is now a molecule which is very intensely investigated. There could be some interesting find as yet, but I think we know a lot about this molecule for the last 30 years.
So let's move on to Avastin and its clinical trials. Initial safety data on a breast cancer trial came back a little disappointing in September of 2002. Were you all holding out hope after the very promising animal model studies, or was it immensely disappointing?
Yes, you're referring to a phase 3 study in breast cancer. Well, actually, it was a patient with advanced breast cancer, relapsed breast cancer. I mean, in retrospect, it was a bit of a long shot, to hope that this drug could. I think the primary endpoint was increasing survival in patients with such a really advanced disease, and it failed, this primary endpoint. But still, in this trial, there was some very intriguing finding because actually there was an increase in response rate. So even though the trial did not meet in that point, it showed there was some biological activity of this drug, At that time, it was very disappointing, but there was really all the success or lack thereof of this program. I kind of was hinging on another phase 3 study in colorectal cancer, previous untreated metastatic colorectal cancer, which was already going on. So we all very anxiously waiting for the result of this trial.
So later, the results from that trial showed a 5-month increase in survival. And the FDA in 2004 approved the use of Avastin for renal cell carcinoma and colorectal carcinoma. So what was your feeling on the day you got the news that it was approved?
It was really stunning. It was an incredible period because there was lots of negative feelings about the angiogenesis. Everybody would have bet that this colorectal trial would have failed. There was even publication from prominent group that actually predicted that the drug that target VEGF is going to fail, just like another class of drugs called MMP inhibitor. Matrix and metalloproteinase inhibitor. So there was actually this sense of inevitable failure of this effort. So it was incredibly refreshing study, see that actually this data could defy these dire predictions and certainly validate a lot of work.
Did you have champagne? Was there a big party? Did you have a parade on the Genentech grounds?
I don't think so. I remember actually I learned the news from actually Arthur Levinson, our CEO. He was so kind, to call me. I was actually in Siena, doing like a week of lecture in this lovely city in Tuscany. I remember I drank a whole bottle of Chianti after I had this news.
Well done. All right, so let's move on to Lucentis, which has been spectacularly successful at not only treating but reversing the effects of wet age-related macular degeneration. How involved were you with turning attention away from tumor angiogenesis to the eye?
I think from the very beginning, I was involved, in the work on the eyes, because it was very clear that, eye was a number of diseases like, ischemic retinal disease, like the diabetic retinopathy, were actually characterized by extensive angiogenesis. Actually, one of the earliest hypotheses of postulated that an angiogenic factor called factor X, was important, in this disease. What are the hypotheses proposed? One of the giants of this field, with Isaac Michelson. And people have been looking for this mysterious factor X, for a very long time, since the 40s, And at that time, there was a lot of interest, especially in the early to mid 90s, in VEGF, because it's induced by hypoxia, which is one of the characteristics of in this kind of disease, And in collaboration with the group at the Josley, we were able to show that actually VEGF is a very highly expressed, present high level in eye fluid of patients with a number of ischemic retinal disease, And then subsequently, we did some animal experiments. It was absolutely striking. It was very compelling. Show that an antibody to VEGF, which actually was the same antibody which works in cancer, also had a very dramatic effect. But at that time, people were very concerned that this animal model were not really predictive. And so, in spite of this, compelling animal data, there was a lot of skepticism.
Well, were you involved during the clinical trials? I can imagine that it must have been tremendously rewarding to watch the data come back.
Well, the data there was a very promising. There is a lot of promising initial data, but the compelling data came, I believe, in 2005 from phase 3 studies with this antibody fragment called Lucentis. It was not actually in the ischemic retinal disease, which was the very initial kind of indication. It was actually in wet AMD, which at that time was the leading cause of blindness. And the data were really, I would say the word, stunning, because, people at most were hoping that you could slow down the vision loss with the VEGF. In this case, this trial showed that maybe in 35 to 40% of patients there was actually an increase in visual acuity. And there was you could prevent severe visual loss in over 90% of patients. So this was really totally unexpected set of data.
Have you ever spoken with any of the patients that have benefited from Avastin or Lucentis?
Yes, I've been very fortunate to meet several of these patients. It was one of the most gratifying experiences.
Right. Now, I mentioned before, and you mentioned as well, after 25 years at Genentech, you took a position as the Senior Deputy Director for Basic Science at the UCSD Moores Cancer Center. First question, why? And second, has your time in industry and at Genentech told you anything or helped you figure out how to get things done?
Yeah. Well, first of all, I think at 25 years is a very long time, actually. And once again, I always, I always have tremendous respect for academia. To the extent I was saying a little bit earlier, decades earlier, I strongly considered joining UCSF, but that was not really the right time, After 25 years, after all this set of works has been completed, been done, I thought actually having a different experience, it could be very refreshing. It could be actually, and there are some people who suggest you should change job every 10 years. I stayed a little bit longer than that, So it was a great opportunity joining UCSD. Actually, there was a very charismatic director, Scott Lipman, who was a really very, very dynamic personality, And I really enjoy a lot, I think this has been a great choice, and And I think my experience at Genentech could be very helpful because it taught me how to do things that which you could, like some level of rigor, for example, in developing a drug, which was something very different from what people typically do in academia. So I believe this experience has been very fruitful.
How are you liking writing grants, though?
Well, I think it's something that you have to do. Is one of the really not unpleasant aspects, but you really need to do. But writing grants, force you to focus your research. I think it's an experience.
You've been lauded with a couple of pretty spectacular prizes, the 2010 Lasker-DeBakey Clinical Medical Research Award, and in the inaugural year that it was given to the Life Sciences, the Breakthrough Prize. What have these awards meant to you?
Well, it's a very humbling experience because I think it's wonderful to be recognized, but at the same time, actually, reflecting the work of many people. I think there are lots of people that contribute to this work. And sometimes they don't appear in this big award. I always feel very grateful to the work done by my colleagues, my collaborators. To me, they were almost family. People in my lab, contributed so much to this success, and I thank all of them, And also, all of this also validate, all of this research, I think, it's very important in science.
What do you see on the landscape for angiogenesis research over the next 5 to 10 years?
Well, the field is, you can argue the field is reached a level of maturity because we understood, we know a lot about, the major mediator of angiogenesis. I think that, from the point of view of basic science, we understand a lot, the work on, not only on VEGF, other molecule like the angiopoietins, like a delta-4 notch, as well as many others, actually. Is enormously expand our knowledge. I think, if I remember, when I started working in this field, that there was virtually nothing was known, very, very little was known. So this is wonderful. From a therapeutic point of view, the field actually it's been wonderful to be able to see this clinical result, which has been very dramatic in eye disease. It's been some very solid, result in cancer. Actually, VEGF inhibitor represent, standard of therapy in a number of diseases. But cancer therapy is always very complex. What we need is to find some very effective combination of drug, combination. And for example, there is lots of promise that combining anti-angiogenic agents with immunotherapy, could be very effective. This, I think, seems to be a very promising avenue.
Now that you're out of the clinical trials loop at a company, does it give you any pause to say, I want to get back into that?
Well, you don't need to be directly involved in the clinical trial. I think it's because, after all, I was never really somebody directly involved in clinical trial. There is other people who can do this much better than I do. I was always in this side of science. It was of course, lots of fun to discuss the clinical data, to participate in that. In a medical center, like it's probably even easier doing that. Like I'm initiating some studies for some colleagues. At UCSD, but in cancer and eye disease. It happened to be that the Shiley Eye Institute, is next door to the cancer center. So it's a wonderful experience to be able to interact with all the clinicians, and clinical investigators.
One other aspect you weren't necessarily as involved in the development of Avastin and Lucentis was setting the price. Now that you've had a little bit of distance from Genentech, do you have any comment on the sort of hubbub that centers around the fairly extraordinarily high cost of treatment with Avastin or Lucentis?
Well, I think it's an enormous problem, and the cost of the drugs it's a huge issue. It's lots of social and ramification. But what I do, even though I'm not an expert in the price, but very quickly discovered that the high price is not limited to Avastin and Lucentis. All other cancer drugs have a very high price. Sometimes it's very ironic that other drugs, cost even more than Avastin, even when sometimes they invented this greater efficacy.
Right. Have you ever thought about pursuing another career beyond science and medicine?
Well, to tell you the truth, I never. I will. Science is, to me, I don't think I could be able to do anything else as yet.
All right, then let me ask the question a different way. I'm going to press you on this. If you had to do it all over again and you couldn't be a medical doctor or a scientist, what other profession do you think would have intrigued you?
It's very, very difficult to tell, actually. Maybe I would have, I'm interested in ancient history, in classics that I read, for fun. But I don't think I would be able to make a career out of that. I think I'm very fortunate in being able to find, in science, in research, what I really want to do.
All right. Well, thank you so much for joining me today, Napoleon. It was a real pleasure hearing a little bit more about you.
Okay. Thank you very much, Yushima. It's been a great pleasure to talk with you.